What lectures will you be going over today? Like,
Um,
are we starting from lecture one?
yeah, so today we're going to do lectures 1, 2, and 3. So all the ones that they've done up to this point. Then the next review session will be lectures 4 and 5.
Okay, thank you.
No problem. And I should have, y'all should be able to see the PowerPoints. As of right now, all I've kind of done is highlight specific points, something very basic that y'all could have done. Um, as we get into the bacteriology block, I'll start putting up actual like schematics or diagrams that I used to break everything down and kind of organize my thoughts. They were essentially my study sheets. I'll put those up as we get towards bacteriology. So that'll be the later half of this test. And then that will be all of the upcoming test, block 2 test. This test should be a little simpler in comparison. You're not going to have as much material, but once we get to block 2 and 3, that's when we start having to learn organism names and their traits. So then you're going to have a lot on your plate. It's going to be a lot of organisms just for one test. So when we get to that, that's when I'll start uploading those. So that way you have those and it's much easier to study. All right, so. Alrighty. All right, so the first lecture was pretty introductory. Actually, the first 2 lectures are pretty introductory. They're just kind of going over the immune system. It's at this point it's a 20,000-foot view, so there's nothing too detailed that they're going to be asking you. Mainly, some of the big things are going to be some of the separations. Give me 1 second so I can actually see the chat while I'm doing this. Okay. Can everyone see the slides now? I just have one. I have the first slide up. Okay, thank you. So yeah, like I said, it's pretty introductory. They're not really going to ask you too many hard questions. What you're going to see from these first 3 lectures are going to be things around the differences between the innate and the adaptive immune system. So these right here, I'm sure the other TAs have told you the objectives. This is 100% where their test questions come from. So if you want to know what information is important, what you need to be focusing on in the lecture, I know there's a lot of slides. Right now, there's only about 40. As you get into it, you'll get closer to like 60 and 80 slides in a lecture. This is where you focus your thoughts. So right here is where you start your study sheets. As you go through the lectures, for this lecture, for instance, what you're going to want to do is really focus on comparing, contrasting adaptive and innate immunity. You know, what is immunity? All these, the points 2 here that she has define, and points 3, the primary and secondary lymph organs. Those are things that you really want to focus on. That's how you're going to make your study sheet. So I said it during the success skills. What I did was I tried to get every lecture and turn it into one sheet. This is how you do that. You essentially build off of your objectives. So for my sheet for this, I'd have half the sheet as a flow chart, comparing, contrasting adaptive and innate. So I'd put everything that I know for adaptive in one column, everything I know for innate, and then on the other side of the sheet, I would start to define those terms, and then I would also give a little graph for the lymph organs. So immunity, most of y'all should pretty much have an idea what that is. It's got 3 parts to it. So we're recognizing self versus non-self. Things that can appear as non-self, any sort of antigen, which you're going to see constantly marked as AG. An antigen could be anything foreign or anything like cancer. So any sort of tumor marker or glycoprotein that you have on your cell surface that your body doesn't recognize that's normally supposed to be there. Any sort of virus that will be releasing virions from the inside of the cell. So anything that you see that's not supposed to see that, not supposed to actually be there. It's also supposed to protect from danger, and recognition in immunity is not enough. We actually need to trigger a response. So you're going to see that we're going to get into it a lot more, and you started to get into it yesterday, how we actually initiate that response and what receptors we need and what cells are involved. Most of her tables for Dr. Pross you don't fully need unless they're a summary table. So this one, where do you see immunity? This one's pretty straightforward. It's kind of common sense. She's not going to ask you any sort of specifics on this relating to immunotherapy or AIDS or anything like that. And then this part of the lecture, I know I could tell in the first lecture day that people were asking a lot of questions. You don't need this historical information. She's giving you these ideas for a backdrop, just so you have an idea of where things started, where things came from. It was the same thing as Dr. Anderson in his lecture, where he had the questions about which researchers came up with what and what technological advancements they came up with. You don't need that. Just go straight off the objectives, the information. We won't need to worry about any of the historical facts. So we can skip. Or you can skip this information to the actual vaccination records. This is just for a frame of reference. And you'll see when you have Dr. Eugen, a lot of our professors will hit home about the issues with anti-vaxxers, and they'll get into that conversation a lot. So this is just kind of to give you that point of view. Same thing with the modern immunology. It's just base ideas. There's not too much you need. And then slide 9 is where we actually start getting into the meat of this lecture. So innate immunity just being your very first line of defense. So it's not specific at all. This is just your generalized immunity. This is going to be with the first time something actually enters and gets past your defenses, the very first time, this is what's going to react with it. So whether it be free-floating antibodies that you already have, any sort of NK cells, macrophage, anything like that, just how it's going to interact in that very first stage. And then adaptive immunity, this is what you're going to actually acquire over time. This is what you're going to be making. So this is the antibodies that you'll be making later on, the B cells and T cells that you have. This is pretty important right here, the blood cells. So knowing which cells fall under which category. The diagram on your right is pretty important. You do need to know that which ones are granulocytes and which ones are agranulocytes. What's the difference between them? She'll get into a little bit more. Also, it's something that she adds later on, but this picture in the top right, this little buffy coat in the middle. So you have when you spin the red blood cell or your blood down, you actually get a coagulant of red blood cells at the bottom, and then your white buffy coat, and then the plasma. It's important to note that the white buffy coat is going to be your white blood cells, so all the cells that we're talking about in immuno, as well as your platelets. And then the plasma is where you're going to see antibodies. That's something that she kind of snuck in on us last year, a little question that she snuck in about the buffy coat. So make sure you know when you spin down a tube of a blood sample, what separates into what. So here, once again, just more.
Or Daniel,
sorry.
How do you like us to ask questions?
You can either you can do it in the chat, or you can just pop in, turn your mic off, and just ask. You can pause me in the middle. This one's going to be a lot quicker, this review session. So.
The lymphocytes, real quick. Can we just go over what specifically those are? Because I'm kind of confused on which cells we're talking about when we talk about lymphocytes.
So lymphocytes are going to be B and T cells specifically. So those are ones that essentially live in lymph organs, but it's B and T cells. Then you can add into that plasma cells, which are just mature B cells. So then your granulocytes are going to be anything that has a granule. So it's going to be neutrophils, eosinophils, basophils, and the macrophage.
Thank you. Sorry to interrupt you.
No, there's no problem. This is going to be, as you can already know, this is our first time doing it this way. The way that the TAs used to do it was in a huge lecture hall, and it would just be raise your hand and ask questions. If anyone needs any clarification on anything or just has a question in the middle that pertains to it, just go ahead and fire off. It's not interrupting.
Hi, sorry. Just in regards to the lymphocytes, in one of her other slides, it said that like the natural killer cells were a type of lymphocyte, like a cytotoxic lymphocyte.
So we're going to get into that. So we'll get into that when we get into that lecture. The NK cells are kind of a different animal. So they are under the umbrella of lymphocytes, but when they talk about lymphocytes, they're referring specifically to the B and T cells. The NK cell pretty much does the same thing as a CD8 T cell, so a cytotoxic T cell, but it does it differently, and it's not around for the amount of time that we need it, and it's not as specified. So we'll get into the NK cells a little bit more, but when they say lymphocytes for any test question or anything like that, it's most likely going to be B and T cells.
Okay, thanks.
No problem. Any other questions?
Yes. You said she had asked sort of a question that had stumped you guys. It was, I think, about the plasma. That's where the antibodies are found, you said?
So it didn't necessarily stump us. It was Dr. Pross likes to ask questions that kind of catch you off guard. She asks slightly more detailed questions with things that she just barely mentions. So with this block that you all have coming up, Dr. Pross, really pay attention to the things that she says. Dr. Eugen, pay attention to what's on the slides. Dr. Anderson, pay attention to what's on the slides. And you'll notice that with some of your other lectures. But for this lecture, the one that she caught us off guard with was the Buffy coat. So on slide, what slide is that? Slide 10, that image on the top right, the composition of whole blood. Just know the specifics that the red blood cells kind of coagulate at the bottom and clump. Then you have your white Buffy coat, which is in the middle, which contains all white blood cells. So not just some, but all white blood cells, as well as your platelets. And then, yes, the antibodies are in the plasma.
Perfect, thank you.
No problem. All right, so like I said, when we're on the innate immunity, it's important to know differentiating factors between innate and the adaptive. So just being things like this is antiviral defenses, immediate reaction. That's the biggest thing, is that it's going to be your immediate reaction, so your inflammation response. So how your body reacts to the first presentation of the antigen. It is for antiviral defense, but there is some antiviral defense later on in your adaptive immunity. The big parts here, natural killer cells. So NK cells are only part of your innate immunity. They're not part of your adaptive immunity. So the things that I've highlighted are just kind of the differentiating factors that you should know. Like I said, try your best to condense it all into one page. This lecture will be a lot easier as we get into longer ones. Like I said, I'll have examples for you all. So adaptive versus innate. Adaptive taking days to weeks for antigen exposure. Usually it's going to be about 5 to 10 days before you have your adaptive immunity response. They are only found in vertebrates. And then this is where you have memory. So your adaptive immunity being where you're creating that memory so that way you don't get sick again. So this is the idea behind vaccinations. So we expose you to the antigen of the virus. And then your body creates immunological memory, whether it be through antibodies or through memory B cells. Your innate immunity, it's the same thing, your first response. Humoral factors being antibodies that you've already made. That's kind of an important terminology that you should know there, and she gets into it. I believe she got into it in yesterday's lecture. It is important to note here, point number 4, that they do rely on each other. That one without the other doesn't actually work, and you'll get into some genetic diseases where you have innate but you don't have adaptive, and the symptoms or side effects that can cause. So then deeper into adaptive immunity, you have 2 different types, active and passive. And I noticed someone did ask the question in the lecture, and it was a pretty spot on question. The plasma that they're doing now, that they just actually approved for COVID, is a passive immunity. So it's more of a quick Band-Aid for the problem. It actually doesn't give you long-term immunity to COVID, which is something that we might see as an issue later on. But as of right now, it is a approved treatment, as you all pointed out. It is important to note the different forms of passive. So anytime a sera is given to someone, so a serum, so passive being a mother and the child, whether it be through the placenta, which will go over that, and which immunoglobulin you get through there, as well as through breastfeeding. And then there are other types of sera. So and I believe Dr. Anderson brought it up, rabies. So if you're questioned for being bitten by an animal that is infected with rabies, they give you an immunoglobulin that is passive immunity. So anytime it's sera or you're just getting straight antibodies, that's going to be passive immunity. Active immunity is going to be when you're making the antibodies or when you're putting up the response. Does that make sense to everyone? That's going to be one of the big factors that they're going to be looking for in this lecture, is the difference between active and passive immunity. So then your primary and secondary responses, this is something that it's not necessarily something that's going to be tested upon, but it's something that you need to know when we get into vaccines and viruses. So this is essentially why when you get the flu shot or when you've been sick with a specific virus, then the second time you get exposed, the reaction is lessened. So just because you get the flu shot doesn't mean you're not going to get the flu. The flu shot is just this primary exposure. So you get the primary exposure from the flu shot, your body builds up an immune response, and then when it comes time for the secondary exposure, you can still get sick and still get infected, but your body and your immune system will react much faster and it'll be much more prepared because you already have built the antibodies.
Hi, I have a question.
Sure.
Would you like consider, so like it's innate and adaptive immunity and then like active and passive is underneath like the umbrella of adaptive? Would like primary and secondary be just like underneath active adaptive immunity?
Yes. Yes, this is only, good question. This is only going to be for active, because at this point, so if it's passive and someone gives you the antibodies, you're not making them. So once the antibodies don't have a long shelf life, so once they give them to you, you won't make more, and then you don't have that protection later on for a secondary exposure. So yes, the primary and secondary response that we're seeing here is only with active, which is under the umbrella of adaptive. All right, and then
Hi.
yeah, go ahead.
Just for clarification, like I remember I got the chickenpox like vaccine when I was younger, but I still got chickenpox, but it was a way more milder form. That's the same thing.
Yes, that's the exact same thing. So the idea behind the vaccine isn't always to give you full protection. It depends on the type of vaccine, and we're gonna get into those into a later lecture on this test block. The idea behind the vaccine is to expose you to the virus, so that way you can build up some immunity, and then when you do get infected a secondary time or a tertiary time, you can bring up the proper response, and then the disease is much more mild. These items to note and the summaries that she puts at the end of her slides are extremely helpful. It's her essentially condensing down her main points. So as we know here, the primary immune response is pretty slow. Like we're saying, when you get a vaccine, it's going to be pretty slow on that first initial exposure. Then the secondary is faster because you have more antibodies, and the antibodies have a specific affinity for the antigen. And the affinity for the antigen is something we're going to get into in the third lecture. So immune dysfunction, this one's pretty important, and we're going to get into these a lot more as we continue, and I believe more so in lecture 5. So the immune system's response to self, so overreaction, so that's actually what inflammation is. Inflammation is not necessarily a good thing. Inflammation is something that we see as an overactive immune response. Cross-reaction, so this is going to be issues where you have things like myasthenia gravis, which you'll talk about in every class, where you have a reaction to certain receptors, acetylcholine receptors in the body. You can also have issues where you'll get certain infections, and you can have cross-reacting antibodies to antigens in yourself. So there are certain strep infections that you can actually, if you have them for long enough, your body will create antibodies, and those antibodies will cross-react to receptors in your cardiac cells. Immunosuppression, this we're seeing acquired just being HIV and AIDS. Congenital, they'll go through a few genetic issues with those, but those are ones that deal with MHC class 2. Aging issues, the immune response is going to decrease with age. You saw that in lecture 2 where she talked about NK cells as those decrease with age. Stress, you'll go through this in every class as well, but as your stress increases, your cortisol level increases, and then steroids specifically have a direct response on your immune system and decreasing your immune system. And then tumors are just cells that have past immunosurveillance. You'll talk about them, and tumors actually have very specific ways of avoiding T cells, so that way they actually don't get destroyed. Changes in immunity.
Hi,
Yeah, go ahead.
I have a question. There was a virus a while back, and I just wanted to know it mainly affected the younger population, and the older population was fine, just because the older population didn't overreact to the actual virus, and so the young people, their immune system kind of went into overdrive and started attacking the body a little bit. What was that? Do you know that off the top of your head?
I don't. That is the same thing. That's actually a very similar thing that's happening with COVID right now that we're seeing, and that's why they're using steroid treatment. They're using dexamethasone treatment.
Okay.
So most viruses actually aren't the direct cause of death. It's your body's immune response to the virus. So you do have issues where your body will. It might have been SARS, which is a close cousin to COVID. Essentially, what's happening is your body is trying to fight off the virus, and it doesn't have a very effective way. So then it increases its immune response harder and harder to try to beat the virus, which then you'll learn inflammation is just causing more and more damage to the body.
Okay. All right. Thank you so much.
No problem. So changes in immunity.
Question.
Yep, go ahead.
When you talk about immunosuppression and congenital being MHC2, will you go a little bit into that?
Yes, yeah, we'll go
later
into
on
that in the
the
third
lecture?
Yep.
Okay.
We'll go over the
I have
differences
questions
between
about that, so I'll just wait.
Yeah, we'll go through the differences between 1 and 2, really easy ways to remember it, and essentially what MHC 1 and 2 or HLA 1 and 2, what they actually mean and what they refer to, because they kind of just throw those terms at you and expect you to know which one's which, and it kind of comes at you a little from the side and you're not too sure. We'll go over that in the third lecture.
Congenital and acquired are both part of, I guess, defects of MHC2, or is it just congenital?
Usually just congenital. There are cases where acquired can be issues with MHC class 2. That specifically is going to be HIV. And you'll get into it more when you get into viruses, how that actually works, but which cells HIV specifically targets to cause immunosuppression.
Okay. Thank you.
No problem. All right, so the changes in immunity, this is just kind of an overall idea. This isn't something that they're going to test on this graph. Usually, like I said, most graphs she won't test on or diagrams for Dr. Pross specifically. I will point out ones in the other lectures that are very, very good to use as a form of reference. This one's just kind of an idea of how your immune system decreases with age, which is something that most of you all kind of have an idea about. And then, like I said, her summary slides are fantastic. They don't give you everything, but they do give you general points that you can then expand on. So use these and the objectives to create your study strategies or your study sheets. All right, so lecture 2. Once again, lecture 2 is a little bit of a review for most people. A lot of this stuff will be review until you get into viruses and bacteria, which will be the end of this block. So the objectives, I can't stress it enough, really, really create your study strategies based around these objectives. This is what they will test on. This is what they're allowed to test on. And this is the focus of the lecture. Where you see immunity.
Is that typically for all classes to focus on the objectives, or would you say just like in immuno?
All classes. So all classes, but then what you're going to notice is, and you maybe have already noticed it with Dr. Muffley, some professors don't put objectives on their slides. But the objectives that is the testable material. That is specifically what they are saying they want you to learn out of the PowerPoint and what they are planning on testing you on. So as a rule of thumb, all classes, but you'll see sometimes, and I'm pretty sure it's only Dr. Muffley who does it, that he won't put up objectives on his actual slides. Every immuno teacher puts up objectives. So for this class, you don't have to worry about it.
Thank you.
Yeah, no problem. So Chelsea, the primary lymphoid and secondary, the primary and secondary lymphoid organs, she actually doesn't go over it in the first lecture, even though it is an objective. She covers it in, I believe, this one, where it's going to be bone marrow and thymus and spleen, where she talks about maturation. Last year and the years prior, Dr. Pross wasn't the one who actually gave that very first lecture. It was Dr. Eugen, but he wasn't able to give it. So she kind of took his objectives and then didn't fully adjust them. But she does go over the primary and lymphoid organs in this one and the third lecture. So they are not the primary and secondary responses. They're specific organs where we see lymphoid development. So where do you see immunity? Reactions to pathogens, vaccinations, rejection of foreign grafts, defense against tumors, and then reactions to self. This is something that we're going to go over at nauseum in the next hour or two, and then for the next week. So just a review on adaptive and innate. This is information that you should have had in your study sheet for lecture 1. Just stuff to make sure. Notice that cytokines and the complement system do appear in both adaptive and innate. So that is something pretty important to note, is that you see cytokines and the complement in both. Just to note, cytokines are any sort of protein chemical that we're using that can either stimulate the immune system or have a response, an immune response. So we're going to talk about them a lot: IL-1, IL-6, IL-2, TNF, and then we'll go through them. They never actually want you to know specifically what each one does, but just to be familiar with those names. It's important to note also adaptive or acquired is antibodies and memory. That's the biggest thing. That is the largest differentiating factor. So again, here we see that little diagram, the composition of blood. You definitely need to have that in the back of your mind, because she did throw us that curveball last year that no one was expecting. Innate immunity, an overview. Once again, you've seen this. You saw this in the first lecture. It's fast, within seconds to hours. It occurs, and it's going to be your phagocytic cells, your NK cells, so your macrophage, your dendritic cells, your NK cells, and your neutrophils. So once again, yep, the immune cells are going to be the macrophage, the neutrophils, and NK cells. Make sure you know which cells are involved in which type of immunity. So it's important to know that these 3 are going to be your innate immunity, and then your acquired or adaptive is going to be T cells, B cells, things like that. How an intact barrier works, so that's essentially just your skin or your epithelium protecting you. It is important to know that you do have intraepithelial lymphocytes, so lymphocytes that are within that epithelial system that help you create an immune system or a defensive barrier. And then your cells of your innate immune system, like I said, neutrophils, macrophage, dendritic cells, and NK cells. Make sure you know what differentiates them all. Also, she kind of throws this acronym out there at you, PMNs. So PMNs are just neutrophils. She'll kind of throw it out there later on and talk about it a lot, but PMNs are neutrophils. So they are the most abundant. So at any point when you have an immune response in the very early stages of the immune response, you're going to have a huge number of neutrophils. And that's just your body reacting towards that. It's creating more neutrophils to deal with that antigen or that foreign substance. Okay.
Daniel,
all
I
of
had a
these
quick
do.
question.
Sure, go ahead.
So when she was going through the lecture, she said that neutrophils, like she said that they didn't have nuclei, but then when she was talking about the picture, she was talking about the nucleus. So I was kind of confused. Like,
she probably misspoke. So the neutrophils actually have more than one nucleus, and that's why they're called PMNs. It's a polymorphic nuclei. So it's a polymorphic neutrophil. So if you see in the picture on the top right, you can see the one that look like they have misshapen nuclei and a whole bunch of different, like it looks like they have multiple nuclei. Those are neutrophils. She might have misspoke. The only cells that we have in the body without the nucleus being the red blood cells.
Okay. Thank you.
No problem.
That is what she was talking about. She had referenced and said it was the red blood cells, and then further went on to describe the PMNs as the nucleated ones.
Thank you.
And can I ask a quick question about the last slide that you were on?
Sure.
Because you had said the intraepithelial lymphocytes. So I just wanted to clarify. The lymphocytes are the B cells and the T cells,
T cells, yes.
as we said before. So would you consider them part of the innate immunity then, or part of the adaptive?
They would be adaptive. So in the picture specifically, that would be a B cell. And you can tell it's a lymphocyte because the nucleus is enormous and you have very little cytoplasm. And the little red prongs that kind of look like tongs on the outside, those are antibodies. So the antibodies just being the actual B cell receptors. So because it has antibodies, that means you've already acquired those antibodies through exposure. So these don't help with that initial infection or that initial exposure. These are B cells that are kind of in those epithelial layers that have been essentially stationed there for future exposure.
Okay, that makes sense. Thank you.
No problem. And then, do antibodies bind differently in a viral infection compared to a bacterial? Yes, very differently. So, and we're going to get over it in the difference between MHC class 1 and class 2 in the next lecture. But the way that antibodies interact with viruses and bacteria are very, very different, and we'll get to it. All right, so
Could
these
you
cells.
go over the previous slide to that? So let's see, I think it's slide 7. Yeah. Lack of specificity and then phagocytizing iron fillings. What does she mean by that?
So the iron filings thing, that was just an experiment that they did to find out that there is no specificity in these cells. So if you put a macrophage or a neutrophil or an NK cell, more so a macrophage, in a petri dish with a whole bunch of iron filings, it will just instinctively go and attack the iron filings because it notices this is something foreign. So what that means is it's essentially going to attack anything that is foreign. It's not targeted. Like our body's not saying, hey, macrophage, go attack that one specific cell. It's just sending macrophages in saying, hey, we have an issue. Send these in. They'll clean up everything. They'll clean up the mess.
Okay, thank you so much.
No problem. So like I was saying, the cells in the innate immune system, make sure you know the difference between them. Macrophages are found in tissues. The dendritic cells found in the external environment, essentially these are APCs, so these are antigen-presenting cells that we find in the skin. The macrophage are antigen-presenting cells that we find on internal tissues, so whether it be in the liver or in the kidney or other organs. These two, the macrophage and dendritic cells, are pretty important. As you can see, they present antigens. We'll get into that in lecture 3 as well as lecture 4, how important antigen presenting is. And then NK cells, she talks about them a little bit here. They are cytotoxic. They call them lymphocytes just because it has a similar appearance to a lymphocyte, but it does use granules, and you'll get into it later. It does use granules to induce apoptosis of viral infected cells and tumor cells. So here, this diagram is almost the exact same thing that you're seeing before. It's just kind of summarizing it for you. It is good to note, though, how some of these kill cells. So under neutrophils, macrophages, and dendritic cells, you see that they use reactive oxygen species, so ROS. That's pretty important, and we'll get into that, I believe, in the 4th lecture of how it actually works. And then the NK cells do something different. They use interferons. So this one, y'all should pretty much know this at this point, how it actually phagocytizes cells. So the bacteria become attached through invaginations. What it is the antibodies on the surface of these, or some of the PRRs will, and the PAMPs will interact with each other. When they do, and we'll go over this in later lectures how it actually works. But they recognize each other, they attach. Once that attachment occurs, then it's sending signals into the macrophage or the phagocytic cell to actually engulf the bacteria or the antigen, and then it puts it in a phagosome. In the phagosome then fuses with the lysosome. Sort of bullet point 3 here is very important, because at this point, that's when you're releasing these enzymes that are going to break down. Once this occurs and they break down the antigen, then we're going to be releasing products from the antigen. So this is what an APC does. This is what a macrophage and a dendritic cell does when they're going to actually present antigens. So they find that bacteria, attach onto it, break it down, and then they release those broken down pieces, and they'll release them later on to B cells, so that way we can form antibodies. A lot of this is just a very large overview leading up to her next few lectures. So the innate immunity, PRRs, the pattern recognition receptors, these are things that our cells have, and they're going to recognize PAMPs. So they're going to recognize specific things that are generalized for certain organisms. So they're going to recognize, for instance, LPS, lipopolysaccharide, which you see on bacteria. We see it on gram-negative bacteria. So we have a PRR that recognizes lipopolysaccharide. So it's not recognizing a very specific bacteria. It's recognizing all gram-negative bacteria that have this lipopolysaccharide. And that way we can react to these faster. We're also going to see ones that react to things like mannose, peptidoglycan, all these other types of things. Toll-like receptors we'll get into a lot more. They are the #1 example of PRRs. We do have other ones, being the mannose receptor, and you'll see those on the diagram that she has later on. These toll-like receptors are the oldest component, so it was one of our first PRRs that we can see genetically. It is something that we see passed down through the germline through most species. So here the PAMPs, like I was saying, the LPS, so lipopolysaccharide, is a PAMP that we can see it. So it's something that's just on the surface, whether it be a glycoprotein or any sort of protein that we see on the surface of an organism that lets us know this is foreign. So these are the things that we actually see as foreign in our immune response. So here,
Are the PAMs specific to bacteria?
not necessarily
Are the PAMs
specific to
for bacteria? Sorry,
So
LPS.
So the LPS is specific for bacteria, but all PAMPs are not necessarily specific to bacteria. A PAMP is just a overarching term of a pattern on the outside of an organism or an antigen that we recognize as foreign, and we have a pre-organized receptor for it. So it can be something on the outside of a virus. It can be certain things. It can be maybe a type of flagella that we see on a virus. It can be a lot of different things. So it doesn't have to be specific for a bacteria, but the easiest ones that we're going to know are going to be the toll-like and the mannose, which are both for bacteria.
Okay, thank you.
No problem. So here, I would make sure to know the toll-like. So the toll-like being the LPS. We don't necessarily have to remember that it's CD14. You'll get into that more, and you'll get into more of the CDs and what they are and what they're actually used for. But it is good to know that the LPS is for the toll-like receptors, which does cause an increase in ROS to kill the microbes. The mannose one is another good one for a different type of bacteria, and it will do this. It will do something similar where it's actually going to cause it to phagocytize and then kill the microbe. So most of these PAMPs and PRR interactions are going to end up in actually killing the microbe. So like I was saying, they do target commonality. So they target anything that is common amongst them. So LPS being something that's common in gram-negative bacteria. Peptidoglycan is something that's going to be common in all bacteria, and it's something that we look for. The PRRs for peptidoglycan are going to be more common for gram-positive. And you'll get into these differences in these different PRRs when Dr. Anderson is going to go through his micro lecture. This part's pretty important, the inflammation. You'll get into it when we get into path. You'll get into it later on in this course. You'll get into it in physio. It is important to know inflammation and the steps to inflammation. So some of the key features that we see is heat, redness, swelling, and loss of function. All of these, the heat, redness, swelling, pain, and loss of function are all caused by these cytokines. So they're all caused by IL-1, IL-6, TNF-alpha. And these cytokines, these three, are released by macrophages and PMNs. So they're released by macrophages and neutrophils. So what that means is when we send in a large amount of macrophage or neutrophils, we're causing this inflammation series. And the whole purpose of it is to actually move cells to that area. So it's to increase the amount of immune cells that we can have to that area. So the heat just being a flushing and the redness being a flushing. So we're dilating the blood vessels, getting more blood to that area so we can get more neutrophils and macrophage to fight whatever antigen is there. And then once a first macrophage or a first neutrophil gets there, it'll release IL-1, IL-6, and TNF-alpha, which will then recruit more macrophage and more neutrophils to that area. Remember, macrophage and neutrophils aren't specified, so they're not targeted defense. They'll attack almost anything, which is how you're going to get the pain and the loss of function. The fever just being a result from the cytokines acting on the hypothalamus. You'll get into this a lot more with which cytokine specifically increases the body temperature, but it is a feedback mechanism to try to help fight whatever organism or whatever antigen that we're seeing. So the way that the vascular tissue responds, we're going to recognize those PAMPs, and then once we recognize those PAMPs, then we're going to have cell migration, and we're going to move those PMNs or any sort of plasma cell, which is just a differentiated B cell, into the area of the stimulus, so that way we can get rid of the antigen and start the healing process. Like I said, it's important to know the signs of inflammation, pain, swelling, redness, and heat.
I had a question about the loss of function. That would be the loss of function of what? Of the bacteria or the antigen?
So the loss of function would refer, when we talk about it here, we're talking about our cells. So wherever the inflammation is occurring, as inflammation occurs and we get chronic inflammation, the macrophage will start to attack our cells or our area. So we'll have so much inflammation that we'll have a loss of function of the tissue or the organ that actually has the issue.
Okay, thank you. Yeah.
No problem. All right, so our process of inflammation. This slide is pretty important, but I'm going to skip. Go ahead.
Yeah, Daniel, just to clarify, where are the PAMPs and the PRRs located? On what cells?
So the PRRs are on our cells, and the PAMPs are on the antigens. So whether it be the virus or the bacteria or the fungus.
I have a question. So PRRs and PAMPs are only a part of the innate immune system and not the adaptive?
Yes.
Oh, okay.
So this process of inflammation, this list is pretty important, but I'm going to skip down to a diagram. She has a diagram here that explains the entire process much, much easier. So not this first one, but this second one on slide 21. So here, this process of inflammation, you're going to have to know this for multiple classes, starts with the very first process is rolling. So what that means is you have throughout your bloodstream, you have these immune cells just kind of rolling throughout the blood. So they're just constantly going on constant immunosurveillance. When we have something occur, so we have some sort of damage on this side, outside of the bloodstream, then we're going to have the macrophage interact with that area. So the macrophage is going to interact using its PRR to interact with whatever antigen it comes in contact with. Once it does, it's going to release cytokines, so TNF-alpha, IL-1. These cytokines will then initiate integrins, so these little blue and red and black little glycoproteins, integrins, to appear on the endothelial wall of the blood. At this point, the white blood cell, whatever type it is, whether it's a leukocyte or lymphocyte, it will attach onto these integrins. So it's rolling, it's rolling, it's rolling. This macrophage is going to release the cytokines, initiate these integrins being produced. These integrins are essentially calling this leukocyte or lymphocyte to attach. Once it attaches and it adheres, it's going to adhere completely. So here we have rolling, then your integrin activation, and then your adhesion. Once you have the adhesion, then you'll have migration. So you'll have the leukocyte or lymphocyte migrating through the endothelial layer, so it's going to squeeze its way through this single layer of cells so that it can actually get to the outside and then interact and deal with the actual antigen. So this is the process of inflammation. This is why you get redness. This is why you get swelling. So if you think about it, when you get a mosquito bite, the needle of the mosquito goes in, and what the mosquito actually does is it releases proteins. And those proteins are to numb that area. As they numb that area, then the macrophage or the dendritic cells will come in contact with that protein, release the cytokines, and then you'll have this flushing. You'll have all of these cells rushing to this area to deal with all of these antigens, so these proteins that are there released by the mosquito or whatever virus might be on the mosquito's tongue. So then all of this is causing that redness, that pain, and that swelling. As we have more cells that come to this area, we're losing the function of this area. Our body's essentially shutting down the function to this area so that way it can deal with the antigen that's there. Then after it's dealt with the antigen, the swelling will go down, these cells will go back the other way, go back into the bloodstream, and then go off. So that's our whole process of inflammation. Make sure you know these steps. You don't have to know the details. You don't have to know specifically TNF and IL-1. You don't have to know, specifically that it's integrins, but just make sure you know the steps, that we have the leukocytes or lymphocytes rolling, and we have integrin activation, adhesion, and migration.
Hey, Daniel.
Mm-hmm.
Could you just go back and explain? So it's like the macrophages come in contact with the protein, release the cytokines, then what else? What happens from there on? Just like the beginning kind of.
So the macrophage, like you said, the macrophage comes in contact with the antigen. When it does, it's then going to release the cytokines, which are going to travel off to this epithelial layer. The cytokines are going to come in contact here, and then it's going to force this epithelial layer to release these integrins. So these little markers on the epithelial layer, these little blue and red markers, it's going to release those. And those are going to call, essentially, those are going to kind of let the leukocytes and lymphocytes know, hey, we need you over here. They're going to call them and attract them. Once they attract them, then they're going to adhere. The leukocytes and lymphocytes will adhere to those integrins, and then they'll squeeze through the epithelial layer and migrate to the other side.
Thank you.
No problem.
And real quick, can you discuss the difference between the cytokines and the chemokines? Because I know she had put that on the other slide, but I was kind of confused with their different roles.
So they almost use them interchangeably. Chemokines are going to be ones that are going to, so cytokines are small proteins that are going to elicit a response. So whether it be to release this biomarker on the outside of your cell, or it's going to be, hey, elicit this response to produce more B cells. Chemokines are going to be ones for direction. So chemokines are going to be, hey, cell, you need to go from here all the way to that side. We need you over there. Hey, cell, hey, white blood cell, we need you in the pancreas. So a chemokine will then chemokine is going to be kind of like Uber, if that makes any sense. And then a cytokine is just going to be like a notification of like, hey, you need to do this.
So does the macrophage release, like at the very beginning there, cytokines and chemokines, or is it different?
No. So the macrophage is going to release the cytokines. It's only going to release the cytokines. The cytokines are then going to interact with the cells in the nearby area, and then those cells will release the chemokines. So the ones that are releasing chemokines in this picture are the endothelial cells, these flat cells.
But then those chemokines also allow the integrins to be put in that layer, so that way the the leukocyte can come in. So
the way that it works, and it's a little out of scope, as in they're not going to ask you this much detail, but so you can know, the cytokines are going to tell these endothelial cells, put the integrins on the outside of your cell surface. Then once you have the integrin, it's going to make the chemokine. The chemokine is then going to attract the leukocyte because it's telling it where to go. And then once the chemokine attracts it, then the leukocyte will bind onto the actual integrins. The integrins, so remember, a cytokine tells you what to do. A chemokine tells you where to go. So the cytokine is telling the cell, hey, put the integrin, so like a flag, on the outside of your cell surface, so that way we have something to grab onto. Then the cell is going to release a chemokine attracting the leukocyte. Once the leukocyte is attracted, then it'll bind to the integrin. Does that make sense?
That makes a lot of sense. Thank you so much.
No problem.
Daniel, I'm just trying to make sure I'm drawing a complete picture. So the PAMPs would be located on the antigen, correct?
Yes.
Okay. And the PRRs are
PRRs are on our cells. So they're going to be on macrophage, neutrophils, and dendritic cells.
got it.
And in this picture, it's not showing the antigen, correct?
It is not, yeah. Yeah, so the antigen would be in the picture above, but this picture above doesn't really go through the process. It's a nice cartoon image, but in the picture above, the little red dots would be the antigen.
Okay, and then the top part of that picture, the bottom one,
Mhm.
the one we're discussing, the top part is going to be the in the blood, correct?
Yes, yeah, that white part is going to be in the blood, and then on the outside, that beige-ish color is outside the blood vessels, so wherever the infection's occurring.
Okay, thank you.
No problem.
Daniel,
Mhm.
you said leukocytes and lymphocytes are in the blood and they go into the tissues. Which lymphocytes are you talking about, or is it just leukocytes?
Leukocytes and lymphocytes. So remember, lymphocytes being B and T cells. And
Okay.
then depending on what is occurring, and this is something we get into in the next lecture, depending on what type of antigen it is, we're either going to recruit B or T cells. And we'll get into that with like an easier way to remember it in the next lecture.
Oh, so inflammation involves the innate and adaptive.
Yes.
Okay.
All right. So far at this point, these are the things we've gone over. And then now at this point, she's going to be talking about the NK cells. So you don't need to know too much about this, about the experiment that happened and how they accidentally found out about NK cells. It is kind of cool, though, considering the person who found them works at Moffitt. You're going to see a lot of our professors. If you've researched any of our professors, they are very, very big names, and they have an insane amount of publications and an insane amount of grants, and they are working on some very cool things. So that's something that should give you a little hope about the program as well. So NK cells, they are cytotoxic. Once again, they call them lymphocytes. For test purposes, you can say it's a lymphocyte, but we can see in the picture it has some granules, and its nucleus is not large enough to be a lymphocyte. They are rare in lymphoid organs. Their entire purpose is to attack infected cells, stress cells, and tumor cells. So they attack cells that have some sort of issue going on with them internally. So the infected cells being intracellular microbes, usually for the most part, this is viruses. There are some bacteria that are intracellular that you're going to get into, but whenever it's a virus, think NK cell. A stress cell, so there's an old damaged cell, and then tumor cell, that's also going to be an intracellular issue because there's an issue with replication of DNA. We do lose them as we increase in age. So this is kind of the theory behind why when you're older, you have a higher chance of getting cancer. This, along with telomeres, are kind of that idea of you've lost a defense mechanism against tumor cells as you age. So right here, this is pretty important to know how they kill. So they have two different forms. They have direct killing and indirect. The direct killing is where it releases perforins and granzymes. What these do is these actually create a hole in the cell wall or in the cell membrane. So the perforins and granzymes actually create a hole in that cell membrane to then force the cell to go into apoptosis. Then the other way is going to be indirect. So they're going to be recruiting macrophages to help phagocytize the cell or whatever's infected at that point. So here, this is where they talk about cytokines. So cytokines just being glycoproteins. You guys should know these: autocrine, paracrine, and endocrine. Endocrine is at a distance, but really it's in the bloodstream. Paracrine is one that's nearby, and autocrine is acting upon itself. We will see examples of autocrine and paracrine when we're talking about B cells and MHC antigen-presenting cells for these cytokines. So just keep that in mind. All right, so the complement system. I saw a lot of people, when she was going through the lecture, kind of freak out about this. This is very straightforward, and they want you to know very, very minimal. It's really a 20,000-foot view. So the alternative complement system is without the action of an antibody. So the alternative complement system can destroy bacteria or viruses without an antibody's assistance. The classical requires an antibody. So what happens is in the classical system, you'll recognize this bacteria. Once you recognize the bacteria, the MHC, or the antigen presenting cell, will recognize that, inform the B cell. The B cell will then release an antibody that will recruit the actual classical complement system. Without the antibody, the alternative system will recognize it along the lines of a PAMP. Once it recognizes that PAMP, it will attack that cell or that bacteria on its own without the need of an antibody. Now, both systems do the exact same thing. Both of them create this hole. So they use the C3, and the C3 zymogen will break down and cause a cascade of breaking down other zymogens, and then it'll get them all together and it'll actually create a pore. Once again, we're creating a pore in the cell membrane. Once we create that pore, then that will induce apoptosis of whatever the organism is. So both of them are working the same way for the same end goal of killing the bacteria, but one is going to do it without the antibody, and the other one's going to do it with the antibody. So once again, so far, these are just ways to make sure that you know and you're keeping up with what the idea is. So the major...
Is it safe to say that the alternative is constantly going while the classical requires some kind of PAMP or pathogen to start it up?
So yes and no. So the alternative is usually on constant surveillance, but it does require a PAMP. It would require recognition before it can actually start the cascade and break down the zymogens to destroy the bacteria. The classical requires some sort of APC, so some sort of antigen presenting cell, to then create the antibody, which will then call it into action. But the alternative is just constantly flowing, but it does require a PAMP.
Okay, thank you.
So Dan, just to sum that up, basically the alternative system also uses perforins, but does it with antibodies, and the complement system of the innate does that without antibodies.
So it doesn't use the perforins. Perforins are specific to the NK cells. What it actually does is each one of these proteins kind of burrow away into the cell membrane until they create a tube, and then that tube starts to leak out the actual the contents of the bacteria, whatever it might be, which then cause it to die. But they both do create that actual pore.
Okay, thanks, man.
No problem. And then, is C3 turn out to be a chemokine? So no. So in this complement system, these zymogens aren't chemokines. They're actually technically not cytokines either. So zymogens just being proteins they're large proteins that get broken down to become activated. So you'll see this when you get to the blood clotting cascade. But a zymogen is just a very large protein that once it's cleaved, it can then activate and then work on the next zymogen. So it starts a cascade. So once C3 is activated, so it's cleaved and it becomes C3A, then C3A will then interact with another protein in the complement system, and then it'll cleave that, and then it'll keep going. Classical system works with opsonized pathogens. So opsonizing is the way it's destroying the pathogens. So the question in the chat, classical system works with opsonized pathogens. So opsonization is just a way that it can actually destroy the pathogen. So that's what it's actually, opsonizing is when it's actually putting the hole into the membrane. So both the classical and the alternative actually go through that opsonization process.
I'm sorry, Daniel. Can you just one more time do a 20,000 level quick
of the I'm sorry, go ahead. 20,000 foot view of what?
Of the two systems, just like the high level things between the two systems.
So base knowledge, what you need to know from these, classical requires an antibody to get started. So the classical system will not initiate without an antibody. The alternative does not need an antibody, so it can initiate and start its cascade. Both systems create a hole in the cell membrane to force that organism to die via apoptosis. That is the 20,000 foot view, as high as I can possibly get it.
Thank you.
The other thing would be the C3. So that that actual complement 3 protein is the one that's shared between the alternative and classical.
Hi, Daniel.
Mhm.
Would the C3A portion count as a chemokine and the C3B count as a cytokine?
No, so they're not actually. Neither one is doing the normal role of a chemokine or a cytokine. So a cytokine is telling a cell to do something, and a chemokine is telling a cell where to go. These aren't actually initiating anything with cells. What these are doing are breaking down proteins. So it's kind of hard to see. Let me see if it's in a better picture. Not really. In the textbook has a really good picture about this. But essentially what's happening is the C3 becomes activated so that you have your large C3 protein. When it's activated, it then one part of it breaks off. That one part that breaks off, we don't worry about it. When it's at the C3A portion, it then goes to another C component. I believe it's C5, but I'm not 100% sure. You won't need to know for the test, but that C3A will interact with that C5, for instance, and then it will cause that one to go from C5 to C5A because it's now activated. And then that C5 will then interact with another one, and then it'll activate it. And then all of them end up going to C9. Once they interact with enough C9s and they activate enough C9As, then we have our pore in the membrane. So they don't actually work like cytokines to where they're initiating a cell to do something. They're actually just cleaving the next protein on down in the line. Does that make sense?
I understand.
Okay.
For question complement system, do we need to know where the C3 comes from? Is it just free flowing in
It's free flowing.
the
Yeah,
blood?
you don't need to know where it's coming from.
Okay. And that's all we need. C3 is just free flowing. We don't
Yep.
we don't
C3
know
is
exactly
free-flowing,
who's
and
making it.
so it's constantly being made by cells, whether it be endothelial cells or so a lot of cells are just making it. It's not one specific cell that its entire purpose is just to put out C3. C3 is just a large protein that's made by cells.
Okay, thank you.
No problem. And to answer the question in the chat, the classical system is technically more specific, yes, because it requires an antibody, and that antibody will bind to whatever antigen, so it does have specificity. All right, so to the major cells of the immune system. So lymphocytes here, you can kind of tell it's a lymphocyte when you see that large nucleus. You won't have any histology or any pictures on this test. This is something that'll come back in the second semester, needing to know that much. So here we're going to explain the lymphocytes. Once again, she's going to have a small narrative to explain how they kind of figured it out. You don't need to know the narrative too much. You don't need to know who did what. It is good to know that it was the bursa of the chicken. The bursa of the chicken would just equate to our bone marrow. And that's kind of the scope of what you need. So we do have two major types of lymphocytes here. So the T cells, which are educated in the thymus, which this is where she's going to talk about the primary lymphoid organ. So the T cells are educated in the thymus, and then the B cells are educated in the bone marrow. So at this point, this is where we're getting into the lymphoid organs. So these are the two primary lymphoid organs, the thymus and the bone marrow. They're primary because this is where we first see these lymphocytes being grown and educated. So T cells are cell-mediated, which means they actually destroy other cells. And then B cells are humoral. Humoral just means antibodies. So our B cells are creating our antibodies, and our T cells are the ones actually initiating cell death. So the T cells are actually the killing ones, and the B cells are the ones that are creating our antibodies. So what is meant by education? Education is just the ability to train these B and T cells so that way they can react properly. And you're going to get into this more, I believe, in lecture 5 and lecture 4 when she talks about issues with reaction. So cells will survive if they react to self. They have to react to a small amount of self. So if they don't react to self at all, then the cell has essentially anergy, which means it's not working. So we need it to react a small amount, but we don't want it to react too much. If it reacts too much, then we have autoimmune disorders, which this is something that we're going to talk about as well. So if a cell in the education, if a cell reacts somewhat to self, then it survives. So it's positively selected. If it reacts too much, then we kill that cell because it's negatively selected, because we want to avoid that autoimmunity. Remember, this is just an overview of it. We will get way more into it as we get down in the actual lectures. So T
Daniel?
cells will produce, yes, go ahead.
Could you re-explain what react to self means? Like, does it just mean that it recognizes itself, or?
So what that means, and she kind of goes into it a little bit more in the next lecture, and we'll go into it more after that. So at the level of the thymus, T cells are there. They're being educated in the thymus. So there are certain cells, and they're called Tregs. We'll get into it, but they present these T cells with parts of self. So essentially, they present them with certain biomarkers that are on the outside of certain cells, and it sees how it reacts. So if it doesn't react at all, that means the T cell is anergy. It has no functionality. It's not working at all. If it reacts a little bit, so if it starts a small immune response, or it reacts by releasing cytokines. So if you present that T cell with a specific antigen from a normal cell surface in the thymus, and that T cell will then release a few cytokines, now it's reacting to self. They're showing a reaction to being presented with that antigen. If you present it with that same antigen and it releases a lot of cytokines, and then it attempts to phagocytize or induce apoptosis to that Treg, that cell that it presented the antigen, then that's autoimmunity. It's reacting too much. So we need to kill that T cell before it gets out and goes throughout the whole body and then causes an autoimmune issue. Does that make sense?
Okay, yeah, thanks.
And we'll go over it a lot more in, like I said, I think it's lecture 4 and 5, where we go over the actual issues with education and the autoimmune issues that we get from that.
Daniel, so would you define education as the ability to train to react to self? Is that a correct definition?
To react. Okay. Yes, it is a correct definition. You'd want to kind of narrow it down a little bit more, that you want to react to a certain level of self.
Okay.
So you want to make sure that it reacts,
not too much.
Right, not too much, because that's when we can really get issues.
Perfect. Thanks.
No problem. So the different roles that the T cells have. So T cells produce cytokines. So remember, cytokines tell cells to do things. So what a T cell is going to do is when it's going to try to kill a cell, it's going to release cytokines that will induce apoptosis in the infected cell. A B cell becomes a plasma cell, which is its fully differentiated form, and then it releases antibodies. The antibodies are then markers that can attract T cells or macrophage to then induce apoptosis or phagocytize. Yes, Gabriella, the positive and negative selection do occur in their primary lymphoid organ. So immune responses, this is very important to know. So immune responses aren't actually going to be occurring in the primary lymphoid organs. So there's just education going on in the primary lymphoid organs. We're not going to have an actual immune response in the thymus or bone marrow. Where we see the immune responses actually occurring, so the action of the immune system, is in the secondary lymphoid organs. So lymph nodes, spleen, Peyer's patches. Peyer's patches are essentially lymph nodes in your intestines, in your small intestines. And you'll go through that later. But mainly know that the action of the immune system doesn't occur in primary lymphoid organs. So you're not going to have a T cell response or a B cell response in a primary lymphoid organ, aside from education in itself. MHC 1 and 2 are not necessarily, we'll get into that in the next lecture. Those are not negative and positive selection. It's a whole different way. It's more of a form of communication. But make sure you know that the action of the immune system occurs outside of the primary lymphoid organs. And then here, secondary tissues of the immune system, so lymph nodes, they're just clusters of the lymphatic system. You do have portions of your lymph node. Make sure to know these. She kind of glossed over them a little bit, but it is pretty important to know that the outer cortex has the B cells, the inner cortex has the T cells, and then the medulla has the actual plasma cells. So make sure you know that the outer cortex is B cells, inner cortex T cells, so you know where in your lymph node each cell resides.
Daniel, when we're talking about immune response that occurs in the secondary organ, we're not at all referring to any tumors or cancers or anything like that.
You can be. So what I mean by, and what they mean by immune response is the immune system responding and reacting to something, and actually initiating an inflammatory reaction or an immune response. So it could be cancer in a lymphoid organ, but also if you've ever had a sore throat, or anything like that, or you have drainage postnasal drip, you'll notice that your lymph nodes, either by your cheeks or in your neck, are actually a little swollen, or your tonsils are swollen. That's because those are secondary lymph organs. So what happens is, in the whole grand scheme of things, your macrophage finds an antigen, a bacteria, eats it, breaks it down, releases the antigens from that bacteria to recruit other cells, starts recruiting cells. Then the antigen pieces that have been broken down get grabbed by a B cell. So you have your APC, your macrophage, that is going to show it to your B cell. Your B cell then goes back to that lymphoid organ. In that lymphoid organ, then it's going to start creating antibodies, and then you're going to have something that she kind of glosses over here. You're going to have clonal expansion. So you'll have an increase of B cells with that antibody, and then that's when you get swelling in those lymphoid organs. So that's the kind of immune response I'm talking about, is when you have something and your B cells and T cells need to divide and grow to initiate immune response. That only happens in your secondary lymphoid organs. That will not happen in the thymus or the bone marrow.
I have a question.
Okay.
So, so they're educated in the primary lymphoid organs, and do they like get transferred to the lymph nodes for storage, if that makes sense? Okay.
Yes. Yeah, they will go from their primary lymphoid organs to where they are supposed to go and reside in the actual lymph nodes.
Okay, thank you.
No problem. Okay, so on the spleen, it is a secondary lymphoid organ. Make sure to know its function. So it promotes an immune response, and it also filters out your blood. So it gets rid of damaged red blood cells. It also will filter out anything that you kind of had in the blood, filter it out, go through it, and then use that as a chance to initiate an immune response. It is important, she kind of glossed over it, but it is really important to know the difference between the red pulp and the white pulp. The red pulp is 80% of the spleen, and that's where you're filtering blood. The white pulp, you have two sections in your white pulp. You'll go over this more in histology, but you do need to know this for this upcoming test, because this was another one of those curveball questions that she threw at us out of nowhere that no one was expecting. The white pulp, you have two sections. You have your PALs, which is your T cells, and your germinal centers, which is your B cells. So just make sure you know those two sections, and that occurs in your white pulp. So you're not going to have an immune response in the red pulp. You can have the action of the immune system occur in the white pulp, but not in the red. And then here, clonal expansion. Once again, she kind of glossed over it because we're going to be talking about it a lot more in the upcoming lectures. Clonal expansion is essentially once that macrophage has come in contact with that bacteria, broken it down, gotten the antigens from it, so it's little pieces of either DNA or any kind of surface receptor, so that way we can recognize that bacteria more, and takes it to a B cell, presents it to the B cell. That B cell then will create an antibody. That antibody on that B cell, we want more of that one B cell. So we're going to do what's called clonal selection or clonal expansion, where we're going to get that one B cell that has that one antibody and make zillions of copies of that one B cell that has that one antibody. We do that so that way we can mount a defense. So it's the whole idea of mounting a specific defense. So if we look at the diagram that she has the four colored cells with the actual little receptor on it. That little receptor, that little box looking receptor on the green, red, blue, and yellow cell, that's going to be your antibody. So that antibody is going to come in contact with that antigen. So because the antigen fits perfectly with the red one, we're going to make more and more of that red cell. That red cell will then proliferate. We'll be able to mount a specific response and then we'll keep that red cell around. Not all of them, some of them. We'll keep them around as memory cells. It can occur with B or T cells. So we'll get into it more. Like I said, she kind of just threw it out there, but they'll get into it more. It does occur with B or T cells. Just depends on what we need.
Daniel, what does she mean by effector cells?
So effector cells are ones that are actually doing the work. So an effector cell is going to be the cell that's actually going to be either releasing the cytokines or directly killing the antigen or organism.
Okay, thank you.
No problem. Okay, and then types of immune responses. What was that? Oh, so Ernest, the oddball question dealt with the red pulp and the white pulp. So red pulp is just where we filter the blood in the spleen. The white pulp is where we actually have the T cells and B cells. So T cells being in the pulls, and then the B cells being the germinal centers. The white pulp is where we can have an immune response. So clonal expression is just to increase specificity of whatever specific cell we need at that time to mount the response. So essentially, it's just creating more soldiers for the battle that we need, and we want specified soldiers. Okay, so the immune response, humoral, this is B cell, cellular T cells. Remember, humoral is antibodies. So our antibody response, our humoral response, the antibodies are also known as BCRs, which is B cell receptors. They are effector molecules because they actively attach onto the antigen and cause something to happen. So whether they're going to attract other things or whether they're going to actually induce something to occur, they do actually do action in the immune system. They are glycoproteins. We'll go through it. The heavy and light chains are linked together. We're going to kind of go through which chain is which. They have here the 5 most common, or the 5 classes of antibodies. It is important to know the differences between them. So IgG is the most abundant. IgA is mucosal, so it's found in all mucosal surfaces. IgG is also the smallest. So that's an important factor to remember, that IgG is the most abundant and smallest. IgA is mucosal, so that you find in your nose, your mouth, in breast milk. IgM is the first one made. So whenever we're making antibodies, we make IgM first, and then we go through a process called class switching, which we're going to go through later, where class switching is where we go from IgM into an IgG or an IgA. IgE is allergies. IgD doesn't do much because it's not free roaming. It's always bound onto a cell. These other ones are kind of free roaming throughout the body. They don't have to be bound to a B cell. But IgE is your allergy one. So here's your actual basic structure of your immune globulin, so your Ig or your antibody. You have the yellow one at the top, that's the antigen binding site. And then you have at the bottom your heavy chain here. This is where it actually binds, where you have the carboxylic acid, the COOH at the very bottom. That's where it actually binds to the cell. So this binds to your B cell here, and then the top part is where it actually binds to the antigen. So make sure you know that difference. This down here, the heavy chain, is called the constant region of the heavy chain. So note that it's the constant region of the heavy chain at the bottom because it stays constant. This doesn't change at the bottom. What does change is your light chain up here, the antigen binding site. That changes because we have different antigens. So we're going to change the chemical composition of the top of the Y depending on what type of antigen we're dealing with. If we're dealing with an antigen that binds perfectly here, that's great. If we're dealing with an antigen that binds differently, then we need to change this top portion here so that it can now bind to that antigen. So this is what's happening when the macrophage brings that small piece of antigen to the B cell. The B cell will then go through different strategies to figure out which antibody can bind to that piece of antigen, and it'll change the top portion here of the light chain so that it can bind with the antigen, and then it'll create more of those. This region down here, the heavy region at the bottom, stays constant. Also make sure to note that the antigen binding region has the NH2, and then the heavy chain portion that binds to your cell membrane has the carboxylic acid.
Hi, Daniel, I have a question.
Mm-hmm.
So when it would change the antigen binding site, would it do that through like post-translational modifications?
Yes. Yeah, and it can also do it pre-translational. So we're going to go through it later on. We already have preset in our DNA. We have preset differences and different antigen binding sites. And what it does is it'll break up those different pieces, and it'll take a piece from this gene, a piece from this gene, and kind of mix them and see which one. It'll do like trial and error and see which one does it. So it can either be post-translational or it can be pre-transcription.
Thank you.
No problem. And you will need to know them later on as FC and FAB. So the antigen binding is FAB, and then the COOH, the carboxylic acid heavy chain, is the FC region. So that's another thing. They will go over it later on, but the bottom of your Y, that's the FC region, and this is the FAB region.
Quick question, because she went into talking about monomers and stuff. Are these antigens, like, is it one peptide chain for both? Because I guess I just always assumed it was multiple peptide.
It is so it is one long peptide chain. You won't need to know that. They're not going to ask you anything like that. They're not going to ask you any biochemistry, but it is going to be one long antigen chain for both that are linked through disulfide bonds.
And into the glycoprotein that they were talking about, is that like a post-translational modification to the antigen binding site, or where is that located?
So you can't kind of see it in here, but it the whole thing is a glycoprotein because it has little glucose monomers attached at different points, and that is post-translational, where it actually binds it, and that kind of helps change the antigen binding site as well. So we can change the antigen binding site by changing the proteins that are here with different interactions, whether it's a basic or acidic protein, or it's a hydrophobic or a nucleophilic, things like that. Or we can change this antigen binding site by adding different sugar moieties. Those different sugar moieties can have it interact differently.
Okay, but the sugar moieties aren't just at the antigen binding site; it's throughout.
Yes. Yeah, they are throughout.
Alrighty, thank you.
No problem. So, question: the disulfide bonds are just that's the hinge points. So you can see in the diagram these little red bars. Those are the disulfide bonds. Those are specific regions that allow for mobility to change the size of the Y and the angle, which can allow us to bind different antigens. Okay, so here this domain structure, you don't really need to know that specifically. You don't need to have this memorized. The Y shape is really what you need to know. But it is important to know that the antigen binding is going to be the ends of the Y, and then your constant region here. It's called biological activity because it's what's actually binding with our cells. You don't need to know this either, but this is a picture of all the different versions. So you can see here the IgM on the bottom left. That's the first one you make. The smallest one being IgG, IgE for allergies, IgA for any sort of mucosal surface. Like we said, it is the B cell receptors. I wrote on here again. So you have your heavy chain here on the inside, and then the yellow part is your light chain. Both of these are variable, which means I can change this portion of the heavy chain and this portion of the light chain to affect antigen binding. This constant region here is what never changes. So this single, just the one bottom part of the Y, that never changes, that stays, and that's what we actually use to bind to the plasma membrane. Okay, so T cell independent and dependent antigens. They produce antigens. Antigens produce only IgM antibodies, and then we require T cells to be able to class switch. This is what I was talking about before. We always produce IgM, and then after that we go through a process called class switching to change it to IgG or IgA specifically on what we need. That requires T cells. Now this slide will go into this way more on another lecture. They're going to have an entire lecture pretty much on this, on T cell independent and dependent. But it's good to know that T cells are required for class switching. So to be able to get different types of antibodies, we do need T cell help. Our primary. Yep,
I have a question about the last slide where you have the domain structure. With the two variable regions, can those switch between light chains and heavy chains? Are they interchangeable? Is that what you?
So they're interchangeable with different heavy chains. So this light chain, we can change this out for other light chain variations, but it's never going to go from a light chain to a heavy chain. If that answers your question.
All right, thank you.
No problem. So our primary and secondary responses, we kind of already went over this. The secondary response is stronger because it has there's more antibodies that we've made because we've already gone through that clonal expansion, gone through the affinity, and figured out which antibody we need for this process. So we've made more of them. So then that secondary response can be a lot stronger and a lot faster. Once again, the whole idea of vaccines. So how do they actually see antigens? We've kind of gone over this: T cell receptors and B cell receptors. We'll go over it a lot more later on. This one, point 2, is probably the most important one, that T cells need antigen presentation. So a T cell cannot work unless it's presented an antigen. That's very, very important, that we can't have T cells just attacking and working. It needs to have formal presentation of an antigen before it can actually initiate. Yes, Daniel, the trend would continue with the third and fourth response. Usually it won't get to that point because by the time we've had a third or fourth response, we've actually created enough immune response that your body, you won't even notice that you've been infected. So like the common cold, for instance, you're not getting reinfected with the same common cold every time. You're getting actual slight different variations in different rhinovirus or coronavirus, things like that, that are infecting you each time. So you actually build up an immunity to protect yourself from that specific strain of rhinovirus. The next time you get a common cold, it's a different strain of rhinovirus. So presenting cells, so antigen presenting cells, these are also known as APCs. They're going to reference them a lot as an APC. It's macrophage and dendritic cells. Macrophage and dendritic cells are the ones that, like we've been talking about, break down the antigen, break into small pieces, and then present it to the T cell. So this is the T cell receptor right here. This is what it's going to look like. So your MHC molecule that's on your antigen presenting cell, it's going to interact with your T cell receptor, and it's going to show the actual antigen right here, present it, and then we'll have a response later on. We'll get into that a lot more as we continue on. All right, so I'm going to try to go through the third lecture quickly because I do have questions at the end that I kind of want to get you all to be able to see. So I do want that to actually be a thing. And the questions that I have for you now are different than the questions that you'll be getting on Friday. Does T cell undergo phagocytosis after antigen present? No, the T cell will not undergo phagocytosis after antigen presentation. It'll actually go through a whole nother system where it'll release cytokines upon itself to then elicit a response. So the T cell won't be phagocytized. So T cell immunity and MHC. So these are the objectives. Like I said, make sure you know them, build your study guides around them. So T cell recognition, we've kind of gone through this already. T cells recognize antigens in the context of self, so they have to understand some response to self. The self that we're referring to is the MHC, the major histocompatibility. This is something that we're going to see on almost all cells. We'll go through which ones are on which cells. The antigen is presented from the MHC onto the TCR, kind of like how you saw in the last picture. So MHCs are just clusters of genes that we see. They help with self versus non-self, and they're part of the adaptive immunity. MHCs do interact with both B cells and T cells. So make sure that you know that. There are two different types of MHCs, and we're going to kind of go through that as quickly as possible. MHC is also
Daniel,
yes.
Sorry, if you're on the third PowerPoint, I don't know if anyone else can see it, but I can't. You're still on the second one.
Yeah, I can't either.
Really?
I see the summary of lecture 2.
Okay, give me 1 second to try to fix that then. Is that better?
Yes, perfect.
Okay.
Yep.
So,
could you go back
essentially
and
slides
do the summary again?
The summary of which one?
Or the learning objectives, I guess. Was that what you were going over?
No, I was down on slide 5. So the objectives right there. I'll go through it again really quickly. The objectives right there. Make sure you know them. I'm not going to go through them. So T cells, they recognize in the context of self. The self we're talking about is MHC, so the major histocompatibility complex. That's just what the antigen presenting cells as well as other cells have to interact with the T cells. So like I said, it's for self versus non-self. It is in the adaptive immunity. Make sure you know that MHC interacts with both B and T cells. So HLA is just the human version of an MHC. It's the exact same thing. We're usually going to be using the term MHC just because it's an overarching term, but HLA is the human-specific version. This is what they use when they're doing transplants. So if you're doing any sort of transplant, whether it be a stem cell transplant or an organ transplant, they're looking at 6 HLA markers. And those markers, they have to see if they're the same. So that way, if they are the same genetically between patients, then there's a less likely chance of getting a rejection of the graft, of the donation.
Hi, Daniel. When you say 6 markers, is that 2 markers for A, 2 markers for B, 2 markers for C?
Yes.
6 markers like for, okay.
Nope.
Thank
yeah, 2 for A, 2 for B, and 2 for C. Yep. And that's what we're going to get to here. So we have 2 markers for A, then some for B and some for C. So we have here that there's 3 different classes. We're really only going to concentrate on HLA class 1 and class 2, also known as MHC class 1 and class 2. And we're going to go through the differences in those. This does show codominance, so very similarly to blood typing, that you have different mixtures. And this is where we're saying the 6 classes and the 6 different products that they look at when they're doing any sort of transplant, that they're going to look at those. They're going to look at the A's, the B's, and the C's to see. There are more than 6 that we look at genetically, but when they're doing these, they found that 6 is kind of a good number to look at to have effectiveness of the transplant. So the relationship between them, like we said, it is critical for the transplant. It's kind of what helps you determine whether or not it's going to react too much to self, because as we've gone through, your immune system has to react a small amount to self, but not too much. So if we get those markers exact, then it won't react too much. Here, we don't really need to know too much about these strains of mice. I highlighted TH1 and TH2 because that's something that's going to be important later on. So just kind of keep that in the back of your head, the TH1 and TH2 types and what they do. This is just the different types of transplants. So syngenic is very similar to an auto transfer. So it's from the same type, and allogenic is from a different type. We'll go over that much, much more when we actually get into hypersensitive immune systems. So same thing, allogenic, just being different. And we already know this, that antigens must be presented to T cells, and we have to use the TCR and the MHC. So then this is how they're actually presented. You saw the picture on the last slideshow was better. This one can look a little hectic. Here, you don't need to know the difference between allo recognition and normal recognition. You just need to know that you have your T cell, your MHC, and they bind together, and then you present the antigen. So this is her idea of where we're going. So here we actually get into the different classes. So I've written on here easy ways to remember this. So class 1. Class 1 is expressed on all nucleated cells. So every single cell, excluding red blood cells, has MHC class 1 on its cell surface. The reason for this is because class 1 is for endogenous antigens. So antigens that are from the inside of the cell. So this is going to be things like viruses or cancer. So these class 1 cells, or these class 1 markers, if we have a viral infection, the virus will produce antigens. Then we'll go through a whole process of actually recognizing that antigen inside, so the endogenous antigen inside the cell, and then we will produce that class 1, that MHC class 1 on the outside surface, which will interact with CD8 cells, CD8 T cells specifically. These CD8 T cells will induce cell death, so they'll induce apoptosis. So this is how you deal with viral infections as well as cancer, by noticing the endogenous antigen. Then we produce that class 1, then it recognizes for a CD8 cytotoxic T cell, which will then induce apoptosis into the cell. So it's important. So what I've written at the top here is kind of the easy way to remember it, that class 1 is for the CD8 cells, and we'll go through another mnemonic. That's an easy way to do it. But class 1 is CD8 cells, which are endogenous. CD8 deal with viruses and tumors. So it's anything that's inside the cell. Make sure you know this shape. Make sure that you know that class 1 has an alpha 1, alpha 2, and alpha 3 domain, and it also has a beta microglobulin domain. So it is important to know this, and it's important to know that the only thing that comes in contact with the actual antigen are the alpha domains. So alpha 1 and alpha 2 come in contact with it, and in this one we have 3 alpha units. It's going to be very different for a class 2. So the class 2 deals with exogenous antigens, so anything that's outside the cell. So anything that's outside the cell is going to be bacteria for the most part, bacteria or fungus. Mainly we're going to be dealing with bacteria. If it's dealing with bacteria, that means it's going to be dealing with B cells. So we're going to have working with B cells and APCs. So class 2 are only on B cells and APCs, so the macrophage and dendritic cells. These are the only cells we find class 2 on. What happens is the bacteria, just like we went through before, the macrophage notices it, breaks down the bacteria, gets the antigen, presents it to a CD4 T cell. This is also known as a helper T cell. We're going to get into this a lot more. Just try to remember 4. So the easy mnemonic people remember is if you multiply the class times the type of cell, you get 8. So class 1, CD8 gives you 8. Class 2, CD4 also gives you 8, but that helps you remember that class 2 goes with 4. I didn't use it very much, but a lot of other people did. So this one's going to work with bacteria. CD4 cells. The CD4 cells are going to initiate the formation of the antibodies. So you remember before how we said that in order to do class switching, so to go from IgM to a different type of immune globulin, we need a T cell. This is the T cell we need. We need a CD4 T cell. That's what we need to be able to do class switching to give us IgG, IgA, IgE. Does that make sense to everyone? So I'll go through it again real quick. Class 1 does CD8 cells, so endogenous, so viruses, bacteria. CD8 T cells are cytotoxic, so they induce apoptosis. Class 2 does exogenous, so bacteria, with CD4 T cells. CD4 T cells allow us to do class switching to give us IgG, IgA, IgE, the different immune globulins. So that is pretty much all of that in a nutshell.
I have a quick question. Is there a reason, I think you said IgM's always made first. Is there a reason why? Is that just because of the pathways or?
That's just the one that's made first.
Okay, thank you.
No problem. Also, like I said before, so know the class 1 receptor, what it looks like, that it has 3 alpha units and 1 beta unit, and the 2 alpha units are actually what interact with the antigen. Class 2 has alpha 1, alpha 2, beta 1, beta 2. What interacts is alpha 1 and beta 1. Make sure you know that. Make sure you know that alpha 1 and beta 1 are the ones that interact with the actual antigen. Class 2 will allow for inducing cell death like class 1, but just in a much, much longer sense. So what will happen is you'll induce, so it's an exogenous antigen, you'll induce the CD4 T cell, which will then allow for class switching. So it makes different antibodies. The antibody will then attach onto the bacteria that's in question, and the antibodies will trigger macrophages to come and destroy the cell, or will trigger neutrophils to come and destroy the cell, or will trigger the classical complement system to come and to destroy the cell. So it just takes more steps. All right, so this is just an image of the different classes. Once again, make sure you know the structure of the different ones, class 1 and class 2. I think the pictures before are much better. So then here, class 1 and class 2. So this is the same thing we have just said. This is everything that we said in a more condensed form. So class 1 is CD8, endogenous antigens, and it induces a cell-mediated response. So T cell, so CD8 T cell response. Class 2 is CD4 positive, so it induces a humoral response. So remember, CD4 T cells, these are T cells we're talking about, help the B cell for class switching for the antibody. So the reason I have here draw this is you're going to need to know the names of these receptors. She hasn't gone over it right now, but you'll need to know it later, that the CD28 and the B7, we'll go over those more, but make sure you know that here with the T cell, the CD8 T cell, you have the CD8, you have class 1, HLA1, and the T cell receptor, and you have CD28 and B7. With class 2, you can see right here, class 2, and the T cell receptor, it's going to attach onto the CD4 with CD28 and B7. We'll go into the CD28 and B7 much later in a lot more detail. So once again, same thing. This is just showing you how it looks when it's presented. So the class 2 goes to the CD4, and the class 1 goes with the CT8. Make sure you know which cell does what. Cytotoxic, so it's going to induce apoptosis. This one is going to allow for class switching of IGs. We kind of went over this already. This is the summary, but make sure you know which ones actually do presentation. So in class 1s, the only one that deal with presentation are the alpha. In class 2, it's alpha and beta. So the T cell receptor in greater detail, it's the same thing. We've gone through the whole thing, the alpha and beta. We won't really get into gamma delta much. We'll get into it briefly later on, but just in a snippet in another lecture. Same thing, we've talked about that. You've seen this picture. You've seen this picture again. This is a slightly better picture because it labels the alphas and the betas. And you can kind of see which is doing what. So how do we make different varieties? We don't inherit them. This is done through education. So the T cell receptors, so the different T cell receptors, we just get through education. Same thing we talked about before. It has to bind slightly to self, but not too much. So this is actually how we do antigen processing. We've been talking about this. So the antigen presenting cell will take in the bacteria, the antigen, break it down, degrade it, and then it will present the small pieces of the peptides. So these peptide pieces are from the bacteria, whether it be from the cell membrane or a cell wall or a flagella that's on the surface, something that we can use that can distinguish this bacteria from something else in our body. We'll break down those pieces of peptides, and then we will actually continue. This is where we stopped yesterday.
Hey, Daniel.
Yes.
So, back, I think, I don't know if it was either the first or second lecture, she had like a picture of phagocytosis of a microbe or something else. And afterwards, she said, like, at the end that when it's broken down, some of those are presented on the cell. Is that what you're talking about?
Yes, that's exactly what we're talking about. So we looked at it. It was that same slide that we looked at before where we talked about phagocytosis and how it actually engulfs it, and then it breaks it down, it puts it in the phagosome, mix it with the phagolysosome. That is this process on slide 29. That is the entire thing right here where we engulf it, we break it down in the phagolysosome, then we get those small peptides and we put them on the MHC. So if we're talking about MHC class 2, we take it from outside and we put it on the MHC class 2, and then we present it to our CD4 cells.
Thank you.
Okay. Someone's saying this is where we stopped yesterday. I don't believe that you all stopped here yesterday. So are you saying this is where you started yesterday?
This is where we stopped.
Actually, where we did stop.
Yesterday?
She
Yes,
said that,
finish.
yeah, she
Yeah,
said
we ran out
that
of
the
time.
rest she's going to go
Okay.
through on Thursday.
Okay. Yeah, yesterday was the only lecture I didn't get to listen to, so that is fine. Do you all want me? Okay, so then since we technically have 15, but we're probably going to go a little over, do you want me to go through the rest of the slides, or do you want to just get to the questions?
I guess in next week's session, could we pick up from here then, if that's the case?
We can, yeah. Yeah, seems like everyone wants to do the question. We can do that. We can pick up right from here. Someone mark this and remind me, because I will most likely forget for next week's, because it's a week away.
Sure thing.
But yeah, someone mark this, and yeah, we'll start at slide 30 next time. Let me get the poll everywhere ready.
I have a quick question when it comes to combining lecture 2 and lecture 3. We talked about clonal selection. When we're referring to clonal selection, is it only the presentation? How do I explain this, or how do I ask this? Is it PRRs and MHC that we see activated in this process? Or is it clonal selections are just PRRs?
So clonal selection occurs at any point where we have an antigen being presented and we are creating a specific response, and we need more cells of that specific response. So at any point, whether it's class 1 or class 2, we can have clonal selection. Clonal selection will mainly occur with B cells. So it'll mainly occur in class 2 when we need to make more immune globulin, but it's just going to be any time that we're getting one specific cell and we're copying it in multiple ways.
Okay,
that
and
make sense?
I'm not getting confused when I'm referring to like, yeah, it makes sense, but I'm not getting like, can PRRs actually cause a clonal selection as well?
Not really. So inadvertently they can, not directly. So they're not going to directly cause a clonal selection. So first what would have to happen is we'd have to have the PR and the PAMP come in contact, and then we'd have to have an adaptive immune response where we're going to actually get a presentation of a T cell. Because MHC class 1 and 2 are different than PAMPs and PRRs.
Yeah. I just wasn't sure that she brought it up in the previous lecture versus this lecture. So I was confused in the sense like, can it be both portions of like both receptors, or is it just only one?
So it's going to be, so the PAMP and the PRR are different. That's just part of our innate immune system. Let me, while I'm explaining this, I'll put up the other. Okay. All right, so can everyone see the poll everywhere on the screen? I'm gonna go with, you should be able to see the poll everywhere. If you can see the poll everywhere, please log into the poll everywhere. I have here at the top, you can either text that number or you can respond on that website. And the question should already be active, so you can enter it. Oh, everyone's doing it. Okay, so the PRRs and the PAMPs are different. Those don't actually elicit that response. What does happen differently is with the MHC class 2 and MHC class 1, those elicit a specific response that will allow for clonal expansion.
Okay.
Make sense?
Yeah, makes sense.
Okay. If I'm not explaining it properly, or if you have another question with it, you can always just send me an email, and I'll try to explain it better. So
thank you.
No problem. So for the first question, it seems like everyone got it right, or at least all the people that answered. So a normal person would actually have, for the antibodies, it would be 5 to 10 days. Don't get tripped up on that. It could take weeks, but in actuality, it'll be 5 to 10 days. So that's our first one. So, next question. Activate. So which binding region is not showing the picture?
Are these questions going to be posted on Canvas?
I don't know if I'll be able to actually post them on Canvas. Dr. Anderson and Dr. Pross didn't really want me to give you all the questions the whole time. So that's going to be slightly problematic. I'll ask them again to see if there's a way that I can. But this one seems to be missing.
On our Poll Everywhere app, we can see the picture.
Oh, you can see the picture?
Yes, I can.
Okay. As long as y'all can see the picture, then you all.
I can't see the picture.
No? Let's see if I can find.
What would happen if you tried full screen?
Let me try.
I mean, the picture just says that the world is consumed with coronavirus, and knowing the mode of infection, answer the following question, if that helps.
That does help. Thank you.
That is exactly what it says, yeah. So I'll try to see if I can get the picture to work next time. Oh, there we go. And it's a little late, but the picture is there.
Daniel.
There's like a maximum number of responses.
Yeah, I don't know how many people are actually voting, but like almost immediately it tells me that the poll is full, and then I can't like answer the question.
Let me see if I can change that setting. Response settings. No. I. I'll have to change it for the next one. It's not letting me change the settings on this one. So for the people that are not able to actually respond, we'll fix it on next time. But that would be the correct answer. The reason that D is the correct answer, so it's coronavirus, so it's a virus. So if it's a virus, that means it's MHC class 1. So MHC class 1 has 3 alpha and 1 beta. Does that make sense to everyone? So A would be MHC class 2, which would be for an exogenous. So that would be for something along the lines of a bacteria. Okay. All right, your next question. Yeah, that lock is really annoying. So for the 24% of people that did get to respond, you are correct. So an extracellular infection is going to be exogenous, which is going to be dealing with B cells and antibodies. T cells are going to deal with an intracellular infection. All right, so the fate of the B cell is going to be the plasma cell and the memory cell and the antibody. So good job for the 76% of people that got it. Remember, the B cell does differentiate into a plasma cell. With clonal expansion, you do get the memory B cell. This is how we can protect ourselves later on, and B cells are the ones that produce antibodies. So what is the natural progression of the immune system? Okay. So yes, it is recognition, activation, and then effect. Don't get confused with the cells roll through the vasculature. That's going to be for inflammation. That's going to be different than just the normal immune response. So how does the complement system destroy foreign antigens? So yes, it is producing a pore in the membrane of the antigen. So it's not going to induce phagocytosis, it will induce apoptosis. Slightly different. So it is LPS. So the toll-like is the PRR. So toll-like is what we have on our cells. The LPS, the lipopolysaccharide, is what's going to be on the antigen or the bacteria specifically. So on the gram-negative bacteria. So that's going to be a PAMP. Remember, PAMPs are on their cells. PRRs are on our cells.
How is peptidoglycan not a PAMP?
So peptidoglycan specifically is the cell wall. So we can't differentiate the cell wall of the organism. So we do use peptidoglycan for other parts of the immune system, but not as a PAMP specifically because it doesn't project from the cell. It's not a receptor that we can kind of see on the outside of the cell.
And flagella, I'm assuming, is the same thing because I thought we could also recognize those.
So so we'd have to recognize specific things on the flagella. The flagella as a whole, because we have cells in our bodies, like sperm cells that have flagella, then we wouldn't be able to just recognize flagella as a whole. It'd have to be a specific type of flagella or a specific moiety on the flagella.
Got it. Thank you.
No problem. The biggest thing with these questions is to know that you have to answer what the question is asking. Try not to think too deeply into it. That was a problem a lot of people had, and you really have to get down to the base of what the question is asking, because for almost all the test questions, you'll have 4 answer choices, 2 of which are obviously wrong, and then 1 that's right, and then 1 that's kind of right. And you need to be able to make that distinction and just answer what they are asking. That is probably the best thing that I can give you for these tests coming up. So yes, the classical system does need antibodies and the alternative does not. Man, that goes quick. So it is the pulls. So remember, the white pulp is the only place where you're going to have immune cells. Red pulp is where you're going to have the blood filtering through. With the white pulp, you have germinal centers, which are for B cells, and then the pulls are for the T cells. So this one, that's going to be kind of tough, unless some people have read ahead. Only going to be if you've read ahead, because this is going to be from the rest of the lecture. All right, so this one is, we're going to cover it again next week, but we know IgE is used for allergies. So that's the one immunoglobulin used for allergies. Something that we'll talk about next time, Th2 is also used for allergies. So people that have high numbers of Th2 usually have very, very high allergy responses. They also have a higher response for CD4 cells, which means that these people are going to be more likely to have high allergies, and they're going to have a weaker reaction or a weaker response to viruses. If you have a stronger Th1 response, you have a stronger response to viruses and tumor cells. So Th1, as a note, is going to be for CD8 cells. Th2 is going to be for CD4 cells. But we'll get into that, like we said, for next week.
Daniel, can you minimize this PowerPoint or the screen a little bit because I can't see the full question?
Yeah.
Okay, thank you.
Does that help?
Yeah, that helps. I can see it.
Okay. So our answer being CD4 positive. So exogenous antigen is going to be outside. So bacterial, that's going to be CD4. CD3 is just the cluster differentiation that is used for the T cell receptor and the B cell receptor to actually bond to the cell membrane. CD8 is going to be endogenous, and CD8 and Th1 are very similar.
Now I can't see the question.
Let me try to go back to the... All right, this is the one we already had.
It's closed.
Yeah, this is the question that we already had. Yep, so that's it. Those are all the questions. Next time I'll definitely try to do a better job with the timing.
Hey, Daniel?
Yes.
Is there somewhere that we'd be able to access exam-style questions like that coronavirus question that you asked for those of us that prefer to learn by actually testing ourselves?
So each Friday you'll have the Q&A session. and the Q&A session specifically is going to be about 12 questions from each subject, and they all should be questions like that. So I'm making as many questions as I can like that, the vignette style that the professors like to use. And each Friday it'll cover that week's material. So my questions for this Friday are coming from these first 3 lectures. And then some of the other TAs, theirs are covering the first 4 lectures because they want to cover everything for the first 2 weeks. But we'll have those every single week. That is the only way we are actually able to give you practice questions. Another good way, it's the books. Some of the books have really good ones. The Immuno book has really good practice questions in it. Sometimes they're a little out of scope, but they do have really good questions, vignette style, and ones that kind of hit the main material.
Professors won't be posting like old exams and
no.
Okay.
No. What we will do though, like the week of the exams, we'll put up practice tests. Each TA will have a practice test specifically for their exam, like for their course. So I'll be putting up a close to 50 question practice test that you'll have. What I would suggest for this course, because this is your last test, and what every single person did, pretty much every single person did last year, is they waited until the last minute to study, and the averages were terrible. The biggest thing you can do is start your study sheets now. So if you start doing your study sheets now, like let's say, today after class, or Thursday after class, work on a study sheet for lecture 1, work on a study sheet for lecture 2. And if you keep up to date with your study sheets, then you can ideally put Immuno on the back burner. And then once the exam session starts, like your whole week and a half of the exams, that's just crazy because you have exam and then lecture right after your exam. And you do need to go to those lectures because you will miss a lot and you do not want to get behind. If you've done all of your prep for Immuno before, then the moment you finish genetics, you can take a break and then review all of your Immuno stuff. Do not wait to start making study sheets for Immuno until after your tests. You need to start doing study sheets now because that is going to be truly detrimental if you don't.
Do you recommend doing that for every class or just Immuno?
I do recommend doing it for every class. It makes the exam block a lot easier. Some of the things I recommend for studying, because it's kind of hard for you all because you all can't have study groups. You can, but you can't. It's not the same. I would suggest going to Home Depot or Lowe's and getting a whiteboard. You can buy like the really fancy ones, but you can just go to Home Depot and Lowe's and get like actual like whiteboard like sheets. And in those whiteboard sheets, you can just have them cut it for you. Get a whole bunch of 3M tape and put it back on the back of the wall and actually like write down things. Like write down your flow charts, write down your diagrams, write down the molecules that you need, write down your pathways that you're going to need for biochem. Like do that. So that way you have your own study room inside your house or wherever you're living. That's probably the cheapest way to do it. But I do recommend doing study sheets for every single class. The way that I made the study sheets slightly different than how other people did. And actually a lot of people ended up using my Immuno study sheets for block 2 and 3. You just try to condense an entire lecture into one sheet, just the front. Color code it, make it look really nice. Handwrite it, I would suggest. And then also handwrite it in blue ink. They've shown that blue ink has a higher chance of staying in your memory. Red ink and green ink don't really stay in your memory, and black does nothing. So if you can, do it in blue ink and just like write an actual sheet. You can do it horizontal, you can do it vertical, whatever you want. I have more examples of my study sheets that I'm going to show you all coming up to block 2, because that's when I really started actually effectively making study sheets. My study sheets for block 1 were just trash, and I didn't really learn how to do it until after that.
Hey, Daniel, I had a question. Do you have any advice for how to study for anatomy?
So anatomy is an animal in its own. Most people haven't taken it, and I hadn't taken it up to that point. What I did was I drew things out, and it's super, super tedious and it's super rough. But if you draw things, the chances of you remembering what it is and what's going to attach to it is really, really high. So what I did was, as you kind of go through like each section, so like I think y'all did the back first, which is kind of crazy that he just kind of threw that on y'all to begin with, because that's that's kind of rough. But like on one sheet, like put the label of the bone or the muscle, draw it by itself, and then describe it. Put its origin, put its both attachment points, what it does, some unique things about it that differentiate it from all the others. And then on the next one, do the next one and do like all one side, all on one sheet. So when you get to like arms and legs, do the upper arm all on one side. So all the flexors of the arm, and then do all the extensors of the arm and kind of group them. If you group them and you do it that way, and then you're going to have to either quiz yourself or write it over and over again on like a whiteboard or something like that, then it'll help retain in your memory.
I have a question about your Friday quizzes. You said that you write 50 questions each week, or is that just the week before our exam?
So the Friday quizzes every week, each TA is going to do 12 questions. And then it's going to add up to 48 essentially. And on Friday, we're going to open up the quiz, and we're going to have it open, I believe, from 11 to 1:45. And you can take it on Canvas at any point. It is timed, so you can use notes, but it's going to be hard to do 70 minutes, 48 questions with actually like looking through your notes. So it's a really good time to like test yourself and test your knowledge and see if the way you've been studying. Then before the exam for each course, each TA will post a practice test. Some TAs, there'll be 25 questions, and some TAs will have 50 questions. It all depends on what our program director allows us to do. But that one is going to be just dedicated for that block, and that's going to be just before the exam. The weekly quizzes are only dependent upon the information from that week.
Okay, that answers my question. So for instance, our first exam is on a Thursday. That TA would post theirs maybe on the Monday or Tuesday prior.
It all depends on when he finishes it and when Dr. Muffley allows him to put it up. So what we'll also be doing is our review sessions will change. So like, because my review session is on a Wednesday, the Wednesday before your anatomy test, I won't be holding a review session. There's no need, because you all are concentrating on anatomy and you're going to be studying for anatomy. So we're going to be moving our review sessions around come test time, so that way it can facilitate you guys having a better review directed towards the test that is upcoming.
Okay, thank you.
No problem. Does anyone have any other questions?
I have a question regarding like the exam day. Like, so for example, the first exam is anatomy on a Thursday, and that kind of like lines up with the class time. But like the next week where genetics is the exam, like at 2:00 on a Wednesday, are we expecting like biochem to be the class after the exam, or is it another genetics section? Because that's
no, it'll be biochem.
Okay, so it would just be the other like
it's whatever whatever the other course is for that day is what you'll have.
And then for like the immuno exams that are on Fridays, like do we take the test, and are you guys still gonna have like a TA session that day, or on those Fridays where we have a test there's not gonna be anything?
The second one. So on those Fridays when you have your immuno test, we won't have a review session. You'll just do your immuno test and then shut your brain off and try to do something fun.
Gotcha. Thank you.
Daniel, I have a question. So when we take the, like, you know how we're going to be in class coming up? So when we have those exams, are we going to be taking them in class, like with our computer, or?
Yes.
Okay.
Yeah, for the people that are going to be, I'm not sure how they're going to do it on like which groups are going to be, whether it's group A or group B is going to be in the lecture hall yet. They haven't told us. But if you are in the lecture hall, you'll be doing it with your computer. And then there's an attachment that you need for Google Chrome. I don't know if anyone has told you all about this, but it's Proctorio, an extension. So on your Google Chrome, try to add the extension of Proctorio, and it essentially kind of like locks out your computer and only allows you to do what's on there. And it locks out your computer, listens to your speaker, and it takes over your webcam. So that way they can kind of see and proctor anyone. And you'll need that extension updated on the day of the exams. Most of the professors should have a Proctorio practice test in their course on Canvas that you can do. But make sure you download that. But yeah, you'll have to bring your laptop. Bring an Ethernet cable if you don't have one, because the Wi-Fi is spotty, and the last thing you want is to be in the middle of your test and your Wi-Fi go out and now your test is invalid. So make sure you get an Ethernet cable also.
You're talking about for the people at home?
No, well, for the people at home, you'll need Proctorio. For the people in the actual lecture hall, you will also need Proctorio. If you're at home, I would suggest the Ethernet cable, so that way if your Wi-Fi goes out. But the people in the lecture hall, definitely 100% get an Ethernet cable.
Thank you.
No problem. And then Ellen asks, the exams are all at 70 minutes. Yes, they are all at 2, and they're 70 minutes. All exams are 50 questions, and they are multiple choice to a point. Some have 4. Some have 4, and then some have 2 questions. Some will have more than 4. It's never fill in the blank or anything like that. So if your laptop does not have a port for an Ethernet cable, you can get an expansion port. So it's something that I have. It's called an Anker, ANKER, and you can plug it into yours. Like for you would not be able to use your iPhone hotspot. You would have to use the actual internet on campus or at home. If you're at home, I guess you could use your iPhone hotspot. But you can get the converters that you can plug an Ethernet into, and then it can plug into your laptop. That's what I had to do because mine doesn't have an Ethernet port either. So I just got an Anker, ANKER, and I think it's a 7-in-1 converter, and it works with it perfectly. All laptops should be compatible with Proctorio. You have to use Google Chrome. That is the only way that you can use Proctorios if you're using Google Chrome. Does anyone have any other questions?
The Friday in between the anatomy lecture and then the subsequent 3 lectures, is there any kind of TA review going on that day? Do we know? Is there anything scheduled?
I'm sorry, which one?
Our first exam is anatomy, which is on a Thursday. The Friday following that, will there be any kind of like review sessions that we know?
Yeah, there will be a review session, a normal like TNAQ, like a Q&A. And at that point, you'll have less than the 50 questions because Justin won't be making any questions because you just had your test. So you'll have questions from biochem, genetics, and immuno. And then also Phoebe will most likely be doing her final review before the exam on that Friday before the biochem test on Monday.
Thank you.
No problem.
Daniel, is it okay if you post even the study sheets that you used for the second exam, but now just to see like a good idea of how they're structured? Because I'm trying to do mine and they're kind of trash. Like you said, they're a little messy and they're not that great.
Yeah, so you might have to do it a few times. I can definitely post some of the ones that I'm going to use at the beginning. And then what I was going to do and what Dr. Anderson wanted me to do is I can post one, and then I'm essentially going to be posting skeletons of the others just so you don't have the exact same study sheet, but you have kind of the same way to organize it. For this one, I would suggest doing, on like one sheet, having half the sheet, a separation between adaptive and innate, and writing down everything you have, like grouping all the information you have on adaptive and innate immunity. And then on another section, T and B cell, everything on T cell, everything on B cell. So everything. So with T cells, you have 2 different types. You have CD8, CD4. And then next to CD8, write everything you know about CD8. And next to CD4, everything you know about that. And then, just keep adding on to it. And it's tough because things are going to overlap between the first lecture and the second lecture with your study sheets, but that's okay. So if you've already covered something in your first study sheet and you know it, you don't need to put it in your second one. So you want to try to minimize as much information that you have on them as possible. Does that make sense?
Yes, thank you.
No problem.
Hi, Daniel.
Yes.
When you were explaining with your initial notes for this class, what was you said there weren't the notes you made initially weren't as good. Was it just not having enough content on the study sheets, or what was the issue you had to overcome?
So it's not that the content wasn't good. It wasn't organized well. So the biggest thing that you can do, and you guys will see as we get to like the actual like species, the actual microbial species, is organization is the most important thing. And I don't mean like being organized like using a folder, using binder, all that stuff. I mean like organizing your thoughts. So for instance, when you get to that point where we have all these different bacteria that you have to know, you need to be able to organize the information on the sheet so that you can, when it's test time, you distinctly remember this is over here. Okay, so all my streptococci are on this side of the sheet, and I have all that information in my head. All of my E. coli are all on this side, and I remember it. So just being able to organize where your mindset was. This first sheet, it had all the information, but it wasn't organized well. It wasn't organized in a way that I could sit there and recall, okay, on the second sheet is when I talked about T cells. And when I talked about T cells, I was also talking about CD4 cells. When I was talking about that was talking about class switching. It was just like condensing notes and not putting a theme to it. And that's the hardest part that I had, was being able to put an organizational system to it so that my brain could follow the tracks and kind of follow where I was going.
Got it. Thank you.
No problem. Would I make a study sheet for lecture 1? Yes. Yeah. Because you don't want to miss any tiny detail that might have been in lecture 1. Even though they repeat the information in lecture 1 and lecture 2, you don't want to miss it. If your study sheet ends up being half a page, that's awesome. That's half a page. It's much easier to study, 10 sheets of notes rather than study, or 10 study sheets rather than study like 30 sheets of notes and all the slides. So definitely just, even though it might seem tedious, make a study sheet for every single lecture.
Do you know what their policy is if something happens to your internet during the exam?
So I'm not too sure. I can ask. I can ask Rose. I'm not too sure, because what they did for us last year when we were in person was they had a written one that they just had us do. And even though we had all at home ones in the second semester and in summer semester, we didn't. To my knowledge, no one ran into those issues. So I'm not too sure what their policy is, but I'll ask Rose and let her know that you guys are curious and to find out what the issue or what the policy might be.
Thank you so much.
No problem.
If you have an in-person slot and you want to take the exam at home, is that possible, you think? Or?
I would have to ask Rose to be sure, but to my knowledge so far, the way everything works is if you have in-person, you can go from in-person to at home, but you can't go back.
Yeah.
So if you have that in-person slot, you can go to doing it at home and then going back. I would suggest doing it in person. The only reason I suggest that is it'll create a routine. So if you are there for in person in class, then over time,
just for the exam I'm talking about.
Right. So that's what I'm saying. So even if I would suggest doing the exam in person, because it creates that routine and it separates you from being at home, and it just creates that routine and that testing environment, which is better there than it is at home. But that's my suggestion. I mean, everyone is kind of different. Do I recommend study sheets over note cards? It's a preference. A lot of people use note cards. The problem with note cards for me and the people that in my study group was it just kind of gets to being regurgitating. And note cards is for like straight memorization. And these tests, what happens with them, like some of your tests will be straight memorization, but a good majority of them are application. So instead of them just being like this, what is that? They're not going to be that. It's going to be more like the COVID question that are like vignette, that it's like, here's a scenario, try to decode the scenario and figure out the information you need. And I feel like you can only get to that ability is if you're creating a study sheet. Because by you creating a study sheet, you've created that organizational system in your head. You've determined what you believe is the important information, and then you've put it in a system that you will remember. And then as you go through it, if you force your brain to work in that process of going from point A to point B to point C, and then figuring it out and relaying it all back to A, then those vignette questions you'll be able to answer like this. If you just do note cards, those vignette ones, you won't be able to go from A to B to B to C. you'll get lost. So you can always try it. Like I say, like I would suggest trying a study tactic now, then doing the Q&A questions that we have on Friday. See how you do on the practice test. If you do well, awesome. That means your study strategy is working. If you don't do well, that means you might have to change your study strategy. It's also important that when you're doing these questions and you see which ones you got wrong, look at them and say, well, was it that I just didn't know the information? Did I not read the question right? What did I do? So that way you can constantly adjust and improve. 3-foot Ethernet cable is fine. Whatever size, whatever length you might need, depending on where you are. If you're taking it in class, 3-foot is more than enough. All right, so we've gone about 20 minutes over. I think that's it. I'll be doing this again in the afternoon, but I'll be doing the exact same thing. And then for anyone else, I will see you all on Friday for the Q&A. Have a good class. Enjoy the rest of your week, and I'll see you all on Friday.